Hi students, I am Dr. A. Sagar Kumar. In this video, I am going to explain you about the genetic code, which is the most important topic in molecular basis of inheritance, and it is a complicated topic also. And many type of the neat questions can be created from this topic. Last year also he has given one neat bit from this topic, but it is simple one. Many complicated questions also can be created, neat questions can be created from this topic, and it is actually students used to feel it as a difficult chapter, difficult topic, but I will make it you very easy, and you can and if you practice for 1 hour, you will be able to give any type of the answer to any type of the question. And the students who are feeling they are having enough knowledge in the genetic code, go and see the last of this video. I give an answer the following, answer those questions. If you are answering all the questions properly without seeing the checkerboard, you are having proper knowledge. Now, whenever the genetic code we are seeing, genetics means carrying of the characters one to another, means expression of the character through the gene, in the genetics. Here in the genetic code, why it is required? One gene, what is gene? Cistron. Cistron is the segment of the DNA which is used to codes for a polypeptide that given in the NCERT book. Polypeptide means protein, and this entire thing given by the Crick, the central dogma he proposed. In that one, what he has told, that is the DNA replicates and forms the DNA, and not only that one, DNA used to replicate, heteroreplication will be taken and forms the mRNA by the transcription method. So DNA replication for the self-replication or heteroreplication for formation of the mRNA, they are having the complementary. They can easy, but the thing comes difficult to explain to the scientists how the mRNA gives the guidance to form the protein polypeptide. That is, protein is a polymer of 20 amino acids with peptide bonds, and there is no complementary between the amino acid and nucleotides present in the mRNA, but they are following the guidance of the DNA in the form of mRNA. Means if you change the changing of the nucleotide sequence will cause the change in the amino acid sequence also in the protein, and a new protein will be formed. There is a lot of evidences are there for that one. But how they are following the mRNA, how the amino acids are following to keep it in the proper order, to arrange it in the proper order to form a protein. That will be explained by the genetic code. That will be explained by the genetic code. What arrangement of the amino acids properly according to the guidance given by the DNA by following some genetic code. That is the genetic code. But how the code is formed, they had seen that is the changing of the sequence of the nucleotides in the mRNA causes the change in the amino acid sequence in the protein. The sequence of the amino acids dictated by the DNA through the formation of mRNA. There is no complementary between nucleotides and amino acids to form protein. There is no coordinate. This is complementary. And next, whenever we are seeing and change in the sequence of nucleotide causes change in the amino acid sequence and the type of protein. That will change, it also will change. Means it is following the mRNA. How it is following? That will be explained by the genetic code. And now, so when they are trying to find out this is the sequence are changing there in the mRNA nucleotide sequence, amino acid sequence is also changing. So there's some relation is there. So what happened? This lead to the proposition of the genetic code that is direct amino acid sequencing in the protein. So this lead means this complication leads. What is the how it is happening? Proportion means how it is. And next will be explaining the genetic code is challenging because 20 amino acids are there and hundreds of the proteins are there that they are changing, and the mRNA is guiding which type of the protein must be performed according to the direction of DNA. Now explaining genetic code is a challenge and needed involvement of several disciplines scientists. Scientists, not only the biologists, whoever it may be, genetics are involved. Because it is related to all the faculty, all the things, because the chemical nature and this thing, organic chemists are involved, and genetics are involved, physics are involved, and all these people involved to find out how the amino acids are understanding the code mRNA. That is will be to explain that one. They want to form a code, but that is the genetic code. Then now if you see, whenever they are doing all these things, the involvement of all the discipline scientists, Gamow, who is the physicist, means a scientist in the physics, proposed the code must be triplet. This is the one of the important, and it is the model which we are following now. So first it is proposed by the Gamow. This question only has given in the last year. And there is 3 nucleotides in the triplet. It is followed by the triplet. The code is triplet. Code means here whatever the nucleotide sequence is present on the mRNA, 3 nucleotides will give one amino acid code. That he told. Why he told like that? Because when they try to the how it is coding the amino acids mRNA, only the nucleotides are 4 types of the nucleotides are there based on the nitrogen bases. They are the whatever the purines, adenine and guanine, pyrimidines, uracil and cytosine. Out these 4 only has to follow. The 4 is to give the code for the 20 amino acids. And if it is double, 2 or doublet, means 2 nucleotides are coding for 1 amino acid, 4 into 4, 16 only codons will come. But amino acid number is 20. And then this Gamow told it is permutation combination based. It is 4 cube, 4 into 4 into 4, 64 codons will come for the 20 amino acids. Gamow, a physicist, proposed the codon must be triplet of 3 nucleotides. This based on permutation combination of 4 into 4 into 4 generated 64 codons for 20 amino acids, which is more than required. Amino acids are 20, codons are 64. So what it indicates? It must be the some amino acids must be coded by more than one codon. Then only it can be explained. First, the scientists and they take and it is become challenge, and it is guided by the Gamow has given this type of the proposal, Then what is the evidence? Evidence given by the Khorana, the code is triplet first. Code is the triplet. That evidence provided by the Khorana. So the codons is triplet by synthesizing RNA molecule in the lab. In the laboratory, instrumentally he prepared. How? Because he taken the RNA nucleotides, separated all the RNA nucleotides and formed in the particular sequence. Means generally the sequence, RNA sequence will be given by the DNA, but in the lab artificially they given, mean they compose one to another, and they prepared artificial synthesized RNA molecule, and this whatever they done, that artificial synthesized RNA molecule codes the amino acids, means bases, three amino acid triplets in the form of that homopolymers or copolymers. What is that one in the checkerboard? I will explain. Homopolymers means UUU, phenylalanine. UUC, that is also phenylalanine. But UCU, that is what it is? Serine. That is copolymer. Like that it is doing. It is proved by the Argan Khorana, and he proved that is the genetic code is triplet. Proposed it is a triplet is proposed by the Gamow, proved by Khorana. How? By forming the RNA molecule, synthesizing the RNA molecule in the lab, instrumentally formed. And now the thing is, one more scientist, that is Nirenberg, cell-free synthesis for the protein synthesis is done. What he done? Already they know how to form the RNA molecule artificially. Based on that RNA molecule, in the ribosome, they taken the ribosomes, free ribosomes, and kept in the media, and he prepared a protein. So cell-free condition, they synthesized RNA molecule. From the RNA molecule, they synthesized protein also. In these experiments, what they came to know, some amino acids are coding more than one codons. That also proved. This is proposed by the Gamow. So Gamow has proposed and proved by the Khorana and Nirenberg. And Nirenberg and Khorana done these things by formation of the basic formation is the RNA molecule. They synthesize. And for the synthesizing means they taken the RNA nucleotides and they jointed one to another by phosphodiester bond, and they used for that purpose Savaro-Wakara enzyme for joining of the two nucleotides. They use the Savaro-Wakara enzyme, that we used to call it as polynucleotide or phosphorylase. Enzyme name is polynucleotide phosphorylase. By using this enzyme, the Khorana and Nirenberg, they done their experiments. That also given in the textbook. This is the thing, but little bit complicatedly given in the NCERT book. Now they came to know that is the codon is the triplet, and some amino acids are coding more than one codon, and both the things was expected by the Gamow. Now the checkerboard is prepared. So finally, checkerboard for genetic code prepared after all the things whatever. Now whenever we are seeing here, now I will explain you these things very easy way how to remember all the things that already I told you in the starting. This is the chart given in the NCERT book. In this one, I given some sequence of the amino acids here. Before explaining this one, you should know one thing. It is a triplet, means 3 nucleotides used to present. 3 nucleotides are present. How the 3 nucleotides will come? In the 3 position, first position, second position, third position. They will take 1 nucleotide from one position, second nucleotide from one position, and one more nucleotide from the third position. Then triplet will be formed. And 4 into 4 into 4, 64 codons has to come. So in the first position, they given the pyrimidines and purines, uracil, cytosine, adenine, guanine. These two are pyrimidines, these two are the pyrimidines, and next whenever uracil and purine, these two are the purines, these are two are the pyrimidines. Remember it. Pyrimidines, uracil and cytosine, and purines are the adenine and guanine. Remember it properly. And in this first row they given separately, and the second row also same thing they given. And third row, in each one, it is to providing one nucleotide, uracil, cytosine, adenine, and guanine. Like that, all these rows are there. Now, how to follow it? Now you see here, one nucleotide from this, and this will have vertical rows and horizontal rows. Vertical rows, 4 vertical rows, 4 horizontal rows. So first position will form the horizontal rows. Second position will form the vertical rows. Now we'll see, if for example uracil from the first position, this is we can call it a cell or box. Uracil, and from this also uracil, if you take U, it will become. The triplet will become U. And this will become phenylalanine. That is the phenylalanine represented by the two triplets. And next, UUA, U from this, and U and C. Means here it is UUU, phenylalanine, UUC also, phenylalanine, both will come, and we used to give it as a phenylalanine 2. Two triplets are coding for the phenylalanine. Remember it. And next will be the another UUA and UUG will represent leucine. This also phenylalanine 2 and leucine will be the 2 in the first box. Now the thing is, first box contains of the 4 triplets. Out of that one, 2 are the phenylalanine, 2 are the leucine. And the second box completely leucine only. Second box completely CUU, CUC, CUA, CUG. All 4 are representing the leucine. So here leucine represented by the 2 triplets, and here 4 triplets. So 6 triplets are coding for leucine. This is most important. Now I will first let me complete this series, the white vertical row. Then I will tell you easy technique also. Next will be the adenine, uracil, and uracil, and adenine, uracil, and cytosine, adenine, uracil, and adenine, uracil, and guanine. Out of these 4, 3 represents leucine. isoleucine 3. Like this you have to practice it. For that why I am telling, I will explain you. Next, AUG. AUG represent methionine. And in the last box in the vertical column in the first row, that is the guanine, uracil and uracil, guanine, uracil, cytosine, guanine, uracil, adenine, guanine, uracil and guanine. All these four will coded for valine. They will be coded for valine. Now you should remember one thing. Easy way to explain this, understand is, how many 1, 2, 3, 4 cells are there? Each having the 4. 4 into 4, 16. Out of 16, they will be distributed like this. First 2 out of these 4, how do you write? You come to know. That is the UUC and UUA, UUG. Out of 4, 2 will be phenylalanine and this 2, and second box completely, that is the, it will become 6. 6 will be the leucine. And here one thing I want to tell. First of all, you should come to know the which box. All these are, if you think the box or the cell, what is giving the code, which box it is, that you should know. That for that also technique I will do. Forget about the third one. If it is U, 2 U's are there, first box, and CU is there, second box, and AU is there, third box, and GU is there, it is fourth box. Later on, third nucleotide used to come. First 2 nucleotides, that is the pyrimidines and purines, all will code for the leucine only. But here 2 pyrimidines and 1 purine will be there. 1 pyrimidine will be giving the methionine, and valine. This all 4 are valine. Now the same thing will be followed here. That is serine 4 plus 2. This box is serine. In the way, same way we have done. Means UC, UCC, UCA, UCG. All 4 represent the serine. But the serine is present here also. 1, 2 more are there. For that only I wrote it here. 4 plus 2. Serine is having 6 triplets codons, and leucine is having 6 codons, and one more arginine is also having 6 codons. That I will explain all the things detailedly, easy to just first listen this thing, and how to follow also I will show you. You will come to know. So next is C and U, and C again C, A, C and G. All these 4 will give the triplets give the code for the proline. That is the proline. Serine will be 4 plus 2, proline will be the 4. And next will be the 309. 309 used to form like this only. This is the entire box is 309. And alanine is the fourth box. Now what you should know, out of 16 here, 4 only amino acids are there. That is serine, proline, threonine, alanine. But serine is having 2 more here. Like that you practice it. So these are 4. These are the 4. Now you see here how to find out box. I told no. Now you see, if AU, GU, UU is there first box in the first row, vertical row, and C with U is there second box, A with U is there third box, G with U is there fourth box. Here U with C, here see next is the C, C with C and U, and C with C, C with C, C with C. Two C's are coming. That then it is the second box. Two C's are coming. It is the second box. Here and AC is coming. That is third box. GC is coming because it is C. GC is coming fourth box. You see the properly and practice it. Now you see here 309 4 and alanine 4. In the examination also, first two triplet, by seeing the first two triplets, immediately it should come in your mind. That is the which box it is, where it is. For example, AC is there. First, ACU he has given. AC. A is the third one, vertical row C is the C, AC, and he has given U. That is the threonine. Where the box is can be found by the seeing first two nucleotides, bases. Now you see here, third one, most important one, that is the, here it is the G. G, whenever we are seeing here, G with U, all four, and GU. First two letters will be the two nucleotides, you see, common. Only changing is in each box is this one. Remember it. In each box, first two letters are same. Means purine and pyrimidines. And only the thing is third triplet will be, the third nucleotide will be changed. Same you see, U, CU, AU, like that. GU, like. Only changing is this order. You have to practice like that. By seeing the first two letter in the vertical row, which box it is. If it is AU, third box. If it is GU, fourth box. Now you see C, we are taking horizontal row. In the horizontal row, vertical, we see CC and AC. And like this you have to follow. And same thing has to follow, A, like this. In this one, third row, whenever we are explained vertically, UA, all are UA. Uracil, adenine. Because adenine is on the top, uracil is here. Uracil, all four comes to uracil and adenine. And all four will come like this. In these four, first two, tyrosine. First two are tyrosine. Second two are stop codon. Means where it is UA. Second means the first box, third and fourth. So third is the A and fourth is the G. AG. Easy to remember. Because you came to know UA and second half it is AG. Like that you have to practice it. Next, whenever we are seeing C and A, CA like this. So here, for see all four are CA, UCAG. First two are histidine, second two are the glutamine. Two subcodons, histidine 2, and glutamine. Glutamine are the 2 in the second box, last 2. And the next fifth one is, that is the asparagine, this one. So here it is, whenever we are seeing asparagine, in the A and A, whenever we are first two are the asparagine, second two are the lysine, and lysine 2. And last it is the seeing GA. First two are the aspartic acid, second two are the glutamic acid. So like this you have to practice. You may think All the amino acids, how to remember? Already you remember the 10 amino acids I will show you, which came in different places. 10 amino acids you are familiar. Those things, if you want to know, I wrote those things also. That is alanine, serine, and that is glycine, alanine, serine. Where given? In the biomolecule structure also he has given. There you practice, no? 3 amino acids you came to know, no? Already it is in our list. And next, there only in the amino acid, that is the acidic amino acid, glutamic acid has given, that you practice. And base amino acid, lysine. And aromatic amino acids, they he given phenylalanine, tyrosine, and tryptophan. So 3, 4, 5, 6 plus 3, 9 came in the biomolecules itself. 10th one is in the histone proteins which form the nucleosome. There he has given the arginine. So 10 amino acids are in your hand because you read in different places. Only 10 more you have to practice. And those amino acids also you have to practice which box it is. First you should know. First two letters. What are the nucleotides? If you, for example, you see here, he given GA. GA means G in the first line, A in the second line, in the second position. Means GA, this is the box. By seeing the GA, you should vertically third one, horizontally last one. Horizontally third one, vertically fourth. Over it is. Like that you have to practice. And next, whenever we are seeing here, fourth, that is the uracil with G, UG, all are UG. First two indicate cysteine. Cysteine, stop codon 1, tryptophan. Now you see here, cysteine 2, stop codon 1. Here two stop codons, UAA, USC, and here one stop codon UGA and UGC tryptophan. Next, come to the C and G. CG, whenever you are seeing all CG, all the codons are showing the codes for the arginine. And next will be, and A with G, AG, first two will be the serine, second two will be the arginine. And lastly, that is the G and G, like this, all shows the glycine. Now the thing is, it will come just a little bit practice. Now any question, any thing he asks, CAA it given. What is that? C is the here. A is the third position. CA, this is the glutamine. And similarly, A is given. A third in the first row, third, and the third row, A, and third one it is AA lysine. First you practice like this. First two letters in the box all are the same. Third nucleotide only will be changed according to the order of the 4. First, in which box how many are there? For knowing that one, I given this one. In the vertical row first, phenylalanine, 2 representing leucine 6 and isoleucine 3 and methionine 1 and valine 4. If you note down these things, You practice this and this. Then total 16. First to this, next 6 leucine, next 3 are the isoleucine. One is the methionine in the third box. And last box, it is the valine, all 4. And next, whenever you are seeing the vertical second column, if the U, whenever you are seeing UC, if the UC, the all will code for the serine. And next CC, all will code for the proline. And AC, all will threonine. GC, all will be alanine. Now, and next third, UA, UC, first two will be the threonine, next two will be the stop codon, and next two, so in the second box, first two are the histidine, and in the second box with CA, that is the second is the glutamine, and next will be the A, and A, it will be the asparagine first two, lysine is the second two. I wrote this here. Threonine 2, first one, stop codons 2, histidine 2, glutamine 3, asparagine 2. This is the third one. Asparagine, this is the asparagine. And lysine here. And aspartic acid, glutamic acid, 2, 2. So here, the amino acids having 2 codons are, here you see, one amino acid tyrosine, histidine, glutamine, asparagine, lysine, aspartic acid, glutamic acid. 1, 2, 3, 4, 5, 6, 7 amino acids are present in the vertical third row. So here fourth row, whenever we are coming, here it is U and G, UG. First two are the UG, UGC 16. And third codon, the triplet, shows stop codon. This 2 and 2, 1, 3 stop codons. And next is the UGG, tryptophan. So here, next C and G, it used to have the 4. All 4 are the arginine. And next will be the serine. First 2 are the serine, next 2 are the arginine. And third one, G and G will be the glycine. Now, question will come like this. How many codons are representing the amino acids which are having one codon only? How many are there? Here, 1 methionine. Here, tryptophan, 2 only. Like that, you have to practice it later on. Next, how many amino acids are having 2 codons? Now you see, already I told 7 are here and 8, and the next is this is the 9. So 9 amino acids are having the 2 codons. What are they? 7 in this, here 1 and here 1. How many are having the 3 codons? Only isoleucine, only having the 3 codons. And next, how many are amino acids are having 4 codons? Here 4 codons, 1, 2, 3, this is 4 and this is the 5. 5 amino acids are having the 4 codons. And 5 codons, how many amino acids are having 5 codons? No amino acid. 5 codons is not represented by any amino acid. 6 codons are present for the some amino acids. They are the phenyl, that is the leucine, serine, arginine 3. Now you see here, it is a 2, 1 it is having the 2, okay, and 2 are having the 9, 2 plus 11, 11 and 3 will be the 1, 10, and this will be the 5, And if you count all the things, it will come 20 amino acids. So, as it is a video, just I am giving the guidance. You keep the book and practice it in this way. If you want, you note down these things here. Means 16 are there. What are they? How it is? Serine, whatever I underline this thing, double time. Serine is coming the second row and arginine is coming upper cell also. So, like that you have to see. Next will be these are the things, saline features. What are the important features it is having? That is, already we discussed, that is 64 codons are present in this one. 61 are presenting 60 are coding for the amino acids, 3 are coding for stop codons, 61 and 3. And stop codons are not, they are not presenting any, coding any amino acid. And next will be the amino acids coding for the more than one codon, hence they coded degenerated. Means one codon it is okay. If the more than one, two only one codon presenting, that coding is having that is the methionine, and next is this is, next one is tryptophan. Here one code, here methionine and tryptophan. Now we'll see. So degenerated means many will be there, not only one, many. The codon is read on mRNA is contiguous, means here there is no full stops and commas in this one continuously. DNA also will be like that only. That DNA you might have read, continuous fashion, there is no full stop and commas. And next, the code is nearly universal because some exceptions are there. Biology it is common. So that is universal. Example, UUU phenylalanine, and some exemptions are mitochondria and protozoans. Small exceptions are there. That is not, means 90% only accuracy in the biology, remember it. So exceptions are there. And now the most important is the AUG methionine. That is, AUG is the methionine which is having one single codon. That it is having the dual function. This is most important bit. Dual function means it functions as initiator codon also. It is one of the amino acid out of 20, one of them. All will be present in the sequence guided by the DNA. But methionine is to present always in the first. For that only initiator codon. Here the bit, how can every protein will start with which amino acid? Will start with which amino acid? Methionine. See how can you create the bit? Methionine is the initiator. Every protein must have the methionine as it is a initiator, that and it will work as the methionine in the series also. So it is having the dual nature. And next, whenever you are seeing, these three are stop codons: UAA, UAG, UGA. And stop terminator codons. Stop codons, what they will do? Terminator. Here he has given, this is important. What are the terminator codon? If you ask, I know the stop codon, but I don't have the terminator codon, means don't say like that, because in the textbook also he has given. It is terminator codon. What is the terminator codon in the checkerboard? Stop codons are called terminator codons. Next, here you see here now. Answer the following. How many amino acids coded with number of codons and their names with genetic code. This only in the starting I told. So amino acids coding with number. For example, I given one model with one codon, two amino acids. What are they? Methionine and tryptophan. Methionine and tryptophan. Methionine, it is having this is the genetic code. Tryptophan genetic code. Like that, you have to fill these things with two codons. How many are there? 7 plus 2, 9. And with four codons, how many are there? 3 plus 2, 5. And with the 5 codons, no, it 5 codons are not coding for any amino acid. And 6 codons, 3. What are they? You write it here, and their code also you write it. So you note down these things and practice it. These are the things about the genetic code. And genetic code, actually, if you one time if you see, you can understand something. See once more time and try to find out. Now I will give once more explanation for the checkerboard. To understand the checkerboard is most important. Now you see here, four nucleotides, four types of the nucleotides. Two are the pyrimidines, two are the purines, AG. Pyrimidines are the top. Like that you remember. Pyrimidines, what are they? Uracil and cytosine. And purines are the adenine and guanine. So it is 4 into 4, 4, 4. These are the 4, these are the 4, and these are the 4. 4 into 4 into 4. Now you see here, and 4 into 4 will give 16 box. 4 into 4, 16 box. Each box contains of 4 triplets. Each box consists of 4 triplets. So vertical row consists of 4 boxes. 4 into 4, 16 triplets used to present. How the 16 triplets are distributed and their names I given here. Here 5 amino acids are present in this row, and first 2 will be the phenylalanine. Like this, you have to note down those things. Not only here, by seeing the textbook also. And in any box, whenever we are seeing, first 2 letters are same. First 2 nucleotides are same. For example, first see A. Only the third nucleotide will be changed. Any box, first 2 nucleotides are same. By seeing that first 2 nucleotides, you can find out the box first. For example, if it is CA is present, C is the second one in the horizontal and vertical also second. So C, a third is the A is the third. C and A, this is the thing. Histidine and glutamine are there. C and A, it came C second one, A third one. The box immediately should come to your mind. How many are there? 2 are there. Histidine and glutamine. First 2, that is the pyrimidines, histidine. Second 2, purines, that is the glutamine. Now it is easy. Like that you practice it. First 2 letters, first letter indicates horizontal. Second letter vertical. And on that box, all 4 first 2 letters are same. So horizontal letter and vertical letter, by seeing that one, you will come to know the which box it is. In that box, whenever you came to know, immediately you will think how many amino acids are there in that one. For example, this AC. AC will come A and C second one, threonine, all 4 are there. So all 4 are the threonine only. Like that you practice it, you will definitely you will get the success. And one more thing I want to tell you, which of the following amino acid is basic, that is in the biomolecules, but he won't give the name, he will give the codon. Okay. Is it possible to you find out? That is the lysine, which is basic. Lysine, where it is? Here it is the lysine. AAA or AAG he will give. So it is important or not? And histone proteins contain the following amino acid for the making the positive or negative charge. He will give the arginine codon. If we create like that, how you will feel? But up to now he is creating, but the day, year to year, it is becoming complicated. And both are present in the NCERT book only. Then is it necessary to practice like that or not? After getting the question, oh, it is very difficult, means no, no chance. You have to practice like that. Just I am telling you, 1 hour you practice it. After that one, every day 5 minutes you see. Later on, every 5-10 days once you see. You will get everything. But without practice, you cannot. But already you came, now is shown that 10 amino acids you came in different places of your previous study. Only 10 new amino acids. What are they? By reading only you will come to know. Where they are, you will come to know. Make it easy and get the success. If you like this video, please give the like mark. And I want to make some more videos in the second year portion for the 2026 people. This it will cover so many these things, means questions. Similar things will be done by me within 5 days. So for 3-4 videos I will do it like this, and I am continuing with the chapter sexual reproduction in the flowering plants. Tomorrow one more video will come. That is the abiotic factors. So all the things, if you want to see, please subscribe the channel. And this is actually very difficult chapter, and slowly listen and follow and repeatedly see and get the success. And don't forget to subscribe the channel.